Okay, I'm going to get started up for the last segment of the class. Okay, so two more components of membranes that we have to talk about. So glycoproteins and glycolipids are also components of cellular membranes. With both of them, a glycoprotein is simply a protein that has a carbohydrate group attached to it. And a glycolipid is the same thing, but it's a lipid that has a carbohydrate attached to it. So when you hear the word glyco, that's referring to the carbohydrate that's attached to either of those molecules. So some of the lipids within membranes have carbohydrate groups attached to them, and then also some of the proteins that are embedded within the membrane also have carbohydrate groups attached to them. Another component of animal cell membranes, as I mentioned earlier, is cholesterol. I did also mention that maybe I misspoke when I said it, but most prokaryotic cells lack cholesterol, so there might be some that have it. We also already talked about the effect that cholesterol has on the fluidity of the membrane. And if you remember, we described it as a fluidity buffer, and we said that its effect on fluidity depends on temperature. And we said at high temperatures, cholesterol decreases fluidity, and low temperatures at low temperatures it increases fluidity. And you don't need to know this detail for an exam or quiz, but this is just showing you how cholesterol orients itself in the phospholipid bilayer. And you can see that a majority of the cholesterol molecule is in contact with the hydrophobic core of the phospholipid bilayer. Okay. So the next thing we're going to talk about is how the membrane acts as a selectively permeable barrier and how it transports molecules from one side of a membrane to another. So can someone I'd already told you this, but what does it mean again to be selectively permeable? So all membranes are selectively permeable. What does that mean? Sorry, can you say that again? I didn't hear you.
Oh, it allows some to pass through it.
Right. So selectively permeable means that it's selective for what it allows to cross it. So some things are able to permeate the membrane. So if it's able to permeate the membrane, then it can pass through. If the membrane is impermeable to a substance, then the substance cannot pass through the cell. And obviously moving things into and out of the cell, one thing it's going to be important for in terms of moving things into the cell is taking up nutrients, and then also getting rid of waste that the cell produces. It's going to be important in terms of that type of transport. And so what we're talking about is how do different molecules make it from one side to the other, because what we're going to see is that some molecules require proteins to move them from one side of the membrane to the other, whereas other molecules do not require that assistance and are able to pass through a membrane on their own. Okay, so in terms of the picture that's on the board here, what they're showing you here is the ability of these different types of molecules to either cross or not cross a phospholipid bilayer. So this would be the ability of those molecules to get from one side of the membrane to the other if a cell membrane was only made out of phospholipid molecules. So if you only had a phospholipid bilayer, it's very easy for things like carbon dioxide, nitrogen, and oxygen to pass through. So they can pass through without any assistance. But then you can see that it's harder for these molecules like water and urea. And then you can see that it's practically impossible. Everything given enough time can pass from one side of the membrane to the other, given enough time. But these things you can just simply think as not being able to pass from one side of the membrane to the other. So what we're going to see is that for molecules like the ones up here, they can simply move in either direction across the membrane without any help. So they're able to freely diffuse across the membrane from one side to the other. In the case of these ones that have moderate permeability, low or very low, we're going to see that there's actually proteins embedded in the membrane that allow these types of molecules to get across the membrane because on their own, they're not able to penetrate the phospholipid bilayer. Okay, and then one of the major determinants of whether the molecule can do this, pass through very easily, or not at all, is how soluble the substance is in the hydrophobic core. So basically that would be saying how lipid soluble is it. So this area consists of lipid, and the lipid solubility of the substance is one of the main determinants as to whether the substance can pass through the membrane freely or the membrane blocks it. So that's all this slide is saying. So the hydrophobic interior is going to be this picture is showing you the same thing that the last picture showed you, so molecules where it's easy to get through, and then molecules where it's harder. This actually is nicer because it defines the types of molecules in terms of, you're going to see that, that hydrophobic molecules typically pass through the membrane very easily, whereas polar and then ions have a much harder time passing through the membrane on their own. And again, as I told you, in terms of that hydrophobic interior, it's going to be the relative solubility of the substance in that interior, in the lipid, that's going to influence whether it can pass through the membrane or not. And what we're going to see is that molecules that are highly lipid soluble, which are going to be nonpolar molecules. So nonpolar molecules, we said, are lowly insoluble or poorly soluble in water, but in lipid solvents, they're highly soluble. And then we're going to see for molecules that don't pass through at all or pass through very slowly, that they're poorly lipid soluble or poorly soluble in this region, and that's going to block them from getting from one side of the membrane to the other. And that's all that this slide is saying. And I do want you to know a few examples for each of them. So at the top here, it's just telling you that highly lipid soluble particles are highly lipid soluble and able to pass through the membrane. So that's going to be molecules that are uncharged or nonpolar. Example of nonpolar molecules that can pass through the membrane without any assistance are oxygen, carbon dioxide, and fatty acids. So what we'll see, or one thing that's apparent from that is that when you breathe, the oxygen that diffuses into your bloodstream and the carbon dioxide that diffuses out of the bloodstream simply passes through all the cells that would be a barrier between the air sacs in your lungs and the fluid of the blood. And then fatty acids are also nonpolar, and they're able to pass through a phospholipid bilayer without any help. Yeah, that's another example. And then for charged particles, so you're talking about ions, and then also polar molecules, they have a low lipid solubility, and they're going to pass through slowly or not at all. And so examples of that, in terms of particles that are going to pass through not at all, basically, would be monosaccharides and disaccharides, so glucose being a monosaccharide, sucrose being a disaccharide, and then ions obviously being charged molecules. You don't have to know any specific ions, but ions themselves are not going to pass through a membrane without any help. So again, any molecule that's poorly lipid soluble is going to have a hard time passing through the membrane. So what we're going to see is when we talk about the different types of transports, things like glucose, sucrose, and ions need a protein in the membrane to move them from one side to the other. So they're only going to be involved in types of transport that involve proteins, whereas your hydrophobic molecules or nonpolar molecules are going to be able to make it across the membrane without the aid of a protein. So there won't be any protein moving it from one side of the membrane to the other. It can do that on its own. Okay. So the different ways of actually moving across a membrane that we're going to talk about in this class are on the board there. So simple diffusion, facilitated diffusion, active transport, and then we'll talk about endocytosis and exocytosis, and then follow also osmosis. And they did introduce all these forms of transport to you in the lab that you guys did last week. You didn't do experiments on all of them, but you did. They did introduce you to them in the introduction. There's a few things that I want to define that I did define for you in the pre-lecture, but I want to define for you before we get into the different types of transport, because if you don't know them, it'll be difficult to understand what's going on. One is what a concentration gradient is. And if you remember back to the pre-lecture, a concentration gradient is simply a difference in concentration between two areas. Is everyone okay with the word concentration? Okay. So a difference in concentration between two areas, plural. So one example of that would be between the inside of the cell or the outside of the cell. So if there's a difference in concentration between the inside of the cell and the outside of the cell, then there's a concentration gradient that exists across the plasma membrane. So for example, if you had a cell and on the inside of the cell, it's not this large, this is way over, but if you have a 2 molar concentration of sodium chloride on the inside of the cell and a 4 molar sodium chloride solution on the outside of the cell, then there's a concentration gradient for sodium chloride. So you have a higher concentration on the outside and a lower on the inside, so that's a difference in concentration between two areas. So that's all a concentration gradient is. It's just a difference in concentration between two areas. And then what we're going to see is that in some forms of transport that we're going to talk about, we're going to say that things move down their concentration gradient. And then in other cases, we're going to see that things are moved against their concentration gradient. Does anyone remember from the pre-lecture what it means if a substance moves down its concentration gradient? So in what direction are things moving if something moves down a concentration gradient? Right, high concentration to low concentration. So in this example, if I tell you that sodium ions, because in solution sodium chloride breaks up into sodium ions and chloride ions, if sodium ions move down their concentration gradient, then they're moving into the cell. Okay, and then if the concentration gradient were reversed, so one example of that is potassium has a higher concentration on the inside and a lower concentration on the outside. So if potassium ions are allowed to diffuse across the membrane down their concentration gradient, then they're going to move outside of the cell. Okay, and then so obviously then what does up a concentration gradient or against a concentration gradient mean? Yeah, low to high. So you're going to go from a low concentration to a high concentration when something is being moved up or against. And then another difference in terms of those two types of movement that we're going to see is that when a cell transports something down its concentration gradient, so from high to low, that type of movement is not going to require any energy from the cell. The cell does not have to put any energy into moving something down its concentration gradient, but if it moves something against its concentration gradient, then you will have to the cell will have to put energy into that movement. And just simply think of that as, obviously, if I asked you to move water down a waterfall, you aren't going to have to put any energy into that. That's going to happen naturally. But if I ask you to move water up the waterfall, then you're going to have to put some energy into moving the water in the opposite direction. Okay, so we're going to see that down a gradient does not require any energy from the cell, but up a gradient does require energy from the cell. And then the second thing that I want to define before we get into some more specifics, which I defined for you in the pre-lecture, is an electrochemical gradient. And an electrochemical gradient is actually the combination of two gradients. Okay, and it is the combination of a concentration gradient. That's the chemical part of an electrochemical gradient. So this is one of the gradients that's involved. So an electrochemical gradient, the chemical part refers to the concentration gradient. And so it's the combination of the concentration gradient and the membrane potential. And the membrane potential is what the electro part of electrochemical gradient refers to. So the membrane potential is actually an electrical gradient. Okay. So in terms of membrane potential, I'm not going to ask you for a definition on a quiz or exam, but you obviously would want to know what it is, given that this is the combination of the two. A membrane potential refers to the separation of charge across the membrane. Oops. Okay, so again, the membrane potential, or this electrical gradient, is referring to a separation of charge that happens across certain membranes. Most plasma membranes, so the plasma membranes of most cells, are going to have a membrane potential that is characterized by a slight excess of negative charges on the inside of the cell and a slight excess of positive charges on the outside of the cell. So basically you get this excess of slight charges just around the inside of the cell that is negative, and then a slight excess of positive charges that are on the outside of the cell. And typically that defines the membrane potential of the plasma membrane. And for this particular membrane potential, this electrical difference is going to favor the movement of positive charges into the cell, and it's going to favor the movement of negative charges out of the cell. Okay, so let's say that you have a higher concentration of sodium ions on the outside of the cell, and this is true for the typical cells within your body. For most of the cells in your body, the sodium concentration, ion concentration is higher outside the cell, and it's low inside the cell. So the movement of sodium across the membrane is affected by the electrochemical gradient. So it's affected by the concentration gradient, which again is just the difference in concentration between the two sides. The concentration gradient favors that sodium moves into the cell. And then it's also affected by this electrical charge difference because these sodium ions are attracted to the negative ions on the inside of the cell. So that also favors the movement of sodium ions into the cell. So you have two things favoring the movement of sodium ions into the cell, both the concentration gradient and the electrical gradient. So the electrical gradient, or the membrane potential of a cell, is going to affect the movement of ions. It won't have any effect on polar or uncharged ions, but it will affect the movement of ions across the membrane. Okay. Do you guys have any questions about either concentration gradient or electrochemical gradient? Okay, so the next thing that we're going to do is the first 4 types of transport that we're going to talk about. The first 2 are going to be simple diffusion and facilitated diffusion. And then the second 2 types are going to be 2 types of active transport. So primary active transport and secondary active transport. And you can take those different types of transport and divide them into two categories. Both simple and facilitated diffusion are categorized as passive transport, and active, so then primary and secondary active transport are both obviously categorized as a form of active transport. This breaks them up, and in terms of the differences between the two, is that passive transport versus active transport. One difference that's obvious is that this one's on the slide, is that passive transport does not require energy from the cell. Whereas in active transport, this requires energy from the cell. So one thing about active transport is that it requires energy, whereas passive transport does not. And then the other difference is going to be the direction of movement. In passive transport, molecules or ions are moved down a concentration gradient. So in both in simple diffusion and facilitated diffusion, things are going to be moving down a concentration gradient. And then it's the opposite for active transport. Molecules or ions are transported against their concentration gradient or up their concentration gradient. And this is obviously connected to whether it requires energy or not. So in active transport, it's going to require energy because you're moving something against its concentration gradient. And then passive transport is not going to require energy because you're going down a concentration gradient. Okay. So then when we kind of look within each, one of the biggest differences between simple diffusion and facilitated diffusion is that simple diffusion does not require a membrane protein. Okay, so it's not going to. So there's nothing involved. There's no protein involved in moving the substance from one side of the membrane to the other. It just simply passes through the phospholipid bilayer. And then the difference with facilitated diffusion is that you have a protein that's embedded in the membrane that we'll call a transport protein. And that moves the substance from one side of the membrane to the other. So facilitated diffusion requires a membrane protein, whereas simple diffusion does not require a membrane protein. And then when we talk about active transport, both primary active transport and secondary active transport both require energy. And then what's going to be the difference between the two is that active transport, like facilitated diffusion, requires a membrane protein. So a membrane protein will be involved in primary active transport and secondary active transport. But in active transport, the membrane protein is going to require a source of energy. So something needs to power the protein so that it can move something against its concentration gradient. And the difference between primary active transport and secondary active transport is what is the energy source that the transport protein uses. So I'll state these things explicitly later on, but in primary active transport, the source of energy is ATP. But in secondary active transport, we're going to see that the energy source that the membrane protein uses is an electrochemical gradient. Okay, so primary active transport is ATP is the energy source, and then the electrochemical gradient is the energy source for secondary active transport. Okay. What is it? It's 8:41. Okay, I'm going to come back to, I just want to, because two of these slides are kind of out of order. and then we'll stop for the day. I'll come back to diffusion and define it for you guys next week. I did define it for you in the pre-lecture, but I'm obviously going to go through and redefine everything. This is just something that we've already talked about, and this is highlighting the fact that this is showing you the membrane potential, and I said that most plasma membranes have a membrane potential around them that's defined by an excess of negative charges on the inside of the cell and positive charges on the outside of the cell. And that electrical difference is going to favor the movement of positive ions into the cell and negative ions out of the cell. And then I already did tell you what an electrochemical gradient is. And if you remember, it's the combination of this electrical gradient, so the membrane potential, and the concentration gradient of the substance that you're talking about. So if you say a sodium ion electrochemical gradient, the sodium ion electrochemical gradient is the combination of the concentration gradient that exists for sodium across the membrane, and then the membrane potential of the cell is also going to influence that movement of sodium ions from one side of the membrane to the other. And then something that I'm not going to ask you to list out on an exam or quiz, but I think it's important to know that electrochemical gradients have a lot of different functions inside of a cell. So I haven't told you this yet, but we'll talk about it in the next topic. But electrochemical gradients and concentration gradients are a form of potential energy, which is stored energy that you can use to do work. And the cell can use this stored energy in the form of electrochemical gradient to do various things. The one that we'll get to in this particular topic is how electrochemical gradients can be used to transport something against its concentration gradient. So we're going to see that something flowing down its concentration gradient or electrochemical gradient can be used to move a second thing against its concentration gradient. And that's going to be similar to the fact that you can use water going down a waterfall to power some other process. So that's essentially what's going to happen in that case. Later on in the course, specifically when we talk about cellular respiration, aerobic cellular respiration and photosynthesis, we're going to see that specifically that hydrogen ion electrical chemical gradients are used to power the synthesis of ATP. So we said that ATP requires, production requires energy, and for the enzyme that produces ATP in those cases specifically uses a hydrogen ion electrochemical gradient to create ATP. And then it's also involved in these other things. Another thing that we won't learn in this course is it's also involved in the ability of your neurons to generate electrical signals. So what neurons are capable of doing is they can rapidly change the permeability of their membrane to certain ions. So what can happen is they can generate these electrical signals. One electrical signal that they're capable of is called an action potential. And this is how they're going to communicate with other cells. What they're going to do is a portion of their membrane will become highly permeable to sodium. Sodium ions, which are positively charged, will move into the cell. That's going to make the inside of the cell less negative. And then what happens is then the membrane closes the proteins that are allowing sodium to enter and then opens potassium channels. Potassium flows out, so then the membrane potential becomes more negative again. So you have this electrical signal where the membrane goes positive and then back down to negative, and that's an electrical signal that neurons and also muscle cells are capable of generating inside your body. And that is going to be produced by the fact that you have electrochemical gradients, because sodium ions flow into the cell down their electrochemical gradient, and then potassium ions flow out of the cell down their electrochemical gradient. So those electrical chemical gradients are the reason that those ions flow in a particular direction to produce an electrical signal. Okay, so those are just some of the uses of electrochemical gradients inside of a cell. Do you guys have any questions so far before I move to the announcements for next week? These are going to be announcements for next week. Some of these I'll repeat in lab this week. And obviously I always send you guys a reminder email on Thursdays. So one thing is that next Monday you have your third lecture quiz. And in terms of this quiz, It's going to cover the part of cell structure and function that was not on the exam. It's also going to cover all of viral structure and reproduction. And then it's also going to cover the membrane structure, synthesis, and transport, but only slides 1 through 12. So basically 1 through 12 discusses the composition of membranes and then their structure, and then we also included fluidity within that. So 1 through 12, so up to and including slide 12 for membrane structure, synthesis, and transport. And that's on Monday. Also next week, you're going to be setting up the experiments for your research project that you turned in a proposal this Wednesday for. And so what we'll do next week is on Monday, we'll start off here, but then we're going to move to the lab, and I'll give you guys lecture time. We're not going to go over. I'll give you guys lecture time to set up the experiments, and then in Wednesday in lab, you're going to have to. What I'll do is place your projects out on a cart, and at some point during the lab, while you're doing the lab, you have to take down your experiments and record your results. The biggest thing for this next week is that what you're going to do this week is you're going to turn in your experimental design, and I'll see what you're doing. If I need to adjust it, I will tell you that in lab. So while you guys are doing lab, I'm just going to check what your independent variable is, and if I can get the things that you need for it by next week. If I can, then that's fine. If I can't, then I'll tell you that and maybe help you guys to come up with a different. You won't have to redo the experimental design. You'll just have to remember that you're not doing the independent variable that you actually suggested. And then in terms of the reagents for the experiment the next week, I provide everything that you'll need except for the seeds. So on Monday, someone in your group is going to have to bring seeds for the research project. And if you're obviously going to have to buy them, and I will reimburse you if you just give me the receipt, or if you just tell me how much they were. You don't have to actually give me the receipt. I'll reimburse you for the seeds. And that's one of the reasons that I was telling you that I would go with either lentils or black-eyed peas because you guys have to get the seeds, and those are super easy to find. You can find them in any grocery store. If you just go to the dried bean section, you get a bag of Goya, black-eyed peas, or lentils. And those have enough seeds to set up the experiment that you're going to set up, and they're also cheap. So that's what I suggest for the experiment. So you will need to bring seeds into class on Monday, and then also you have a pre-lab due on Wednesday, and then finally I'm going to push the lab exam back a week. So it's not going to be next week. It's going to be the week after. The reason that I'm doing that is the first lab exam covers the nature of enzyme action, and we're not going to get a chance to actually discuss that lab until this week. So that's a lot. So you're doing enzyme action this week. Next week we'll discuss it, and then their lab exam will be the following week. So your lab exam will be week 8. Do you guys have any questions about the announcements? Post what? Oh yeah, on Thursday. So on every Thursday during my office hours, that's what I do during my office hours is write up the announcements and send them out. So that'll be coming to you on Thursday. I'll also repeat the seed stuff on Wednesday because that way no one will forget about bringing seeds on Monday next week. Okay, if you guys don't have any other questions, that's it for tonight. I'll see you guys on Wednesday.